This is now the third and the last major step of the central dogma, which is translation, or some people just call it protein synthesis because, in the first place, it's basically us using RNA from transcription to finally get the protein that we want, which is pretty much the end of the central dogma. And in translation, there are a lot of things that we can think of as unique compared to the first two steps, because for the first time, we are now dealing with a process that doesn't start with DNA. Remember, when we were dealing with replication as well as transcription, our starting point was DNA, but we don't have that anymore in this case. So it doesn't mean it's any less complicated. Far from it actually, but it's different, and therefore in this recording I'm just gonna more of discuss the introductory details, the requirements before going on to the actual process. Obviously, we need to have our RNA here, which we will be using for protein synthesis. And here, I just decided to come up with a short strand. Although, if you were watching my previous recordings, you should already know that RNA can be made up of several dozens or even hundreds of nucleotides, or even thousands for the longer ones. So here, it's as if you could see here every three bases that are adjacent to each other. Okay, so three consecutive letters, that is correspond to a certain letter here, which is actually the one letter abbreviation for our amino acids. So basically here I'm trying to say that AUG is equal to methionine, UUU is phenylalanine, UUC equates to phenylalanine, and UGG equates to tryptophan. And I will be talking more about where we got these equivalents later. But the message here is clear that I'm saying that for every amino acid that I want to make for our protein later, I need three adjacent bases. And they're kind of, codes or some kind of um, uh, 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 keys, okay, wherein they convey something. And these three bases that equate to a single amino acid is called a codon. So like codon for code. And do note that codons are uh, dependent on the order. So the way we read it, like AUG, so A is the first letter, U is the second, G is the third, is gonna be significant because, for example, I reverse this, it becomes GUA, then it's gonna be interpreted as a different codon. So here, order matters. Okay. All right. And therefore, let me jump to this. Since order matters, remember that in our RNA, remember it's RNA, we have four bases: A, C, uh, G, and U. No more thymine, right? So A, G, C, and U. So if I consider that I have three bases, and then a codon is three bases, it's a triplet, and I just said a while ago that order matters, then we're dealing here with something called in math as permutations, right? Where again, AUG, for example, is not the same as GUA and is not the same as AGU and so on. So if we're going to convert that into a formula, the permutation that's possible here is equivalent to the number of characters we have raised to the number of things that can be combined at a time. In other words, it's going to be four bases raised to the number of slots, which is three. So doing this formula for permutation, 4 raised to 3 is 64. That means we have a possibility of 64 different codons. So name it, be it AUG, AGU, UGA, GUA, UUU, UUC, UUG, GUU, UGU, and so on. All of those triplets would actually be 64. Out of the 64 codons, it should be known that almost all of them, 61 in fact, are coding. When you say coding, they are equivalent to a certain amino acid, just like AUG is equivalent to methionine, UU is equal to phenylalanine, and so on. And do note that I, uh, I do not have the patience to write all of the 64 here. And if I try to copy paste a picture, that's going to be copyright infringement, I believe. So maybe in the description for this video, I'm going to link you to a complete table, it's usually an 8 by 8 by 8. Sorry, it's a 4 by 4 by 4 table, showing us the 64 different combinations. And anyway, out of those, the 61 are coding. For most part, I don't think there's any need for you to memorize all of those 61. But I think it's important for you to know the three remaining ones out of the 64 because they are non-coding, meaning these three, UAG, UGA, or UAA, do not equate to any amino acid. And you actually call these three as our stop codons for a significant reason, as you will understand later when I discuss the actual translation process. But yeah, we have 64 codons. Now, before I even proceed to discussing the other things here, you should think about this. Like, for example, this is weird because this is the only this translation process is the only process wherein we have nucleotides as our starting point. RNA, right? In translation, we start with RNA. It's made up of nucleotides, and then we convert it into a protein, which is made up of amino acids. And that's kind of weird because the moment you cross from the language of nucleotides, which include these, into the language of proteins, there's some, supposedly some intrinsic misunderstanding because these two things are not the same. It's like saying the language of nucleotides is not the same language as amino acids, which, by the way, gives life and meaning to the reason why we use the word translation. Translation because we translate the nucleotide language into the amino acid language. And thus, shouldn't it make sense that we should have some way to make the RNA understand the protein or vice versa? Okay, because basically, if we think of RNA as being magically converted into a protein, it's like in real life you're talking to a stranger with a different language, and then you expect that subtitles would come out from his or her mouth. And of course, that doesn't happen. In that case, we have to find a way to serve as a bridge between our messenger RNA and the protein. Some kind of friend that we have that could understand and translate the two languages for us. Now, in the molecular level, this friend of ours, which we can assume would translate these two languages, is a different type of RNA called the transfer RNA or the tRNA. And we will understand later why I equated this to some kind of person who understands two different languages. You should first know that in the detailed form, the tRNA is actually a single mRNA, not really mRNA, but a single RNA strand, which has some kind of distorted T shape. Wherein here at like one of the ends, we have a set of three bases, which we call as the anticodon. And as the name implies, the anticodon is something that should match the original codon. So think of it this way. Imagine that I have an mRNA strand laid down somewhere, so like this, and then it continues somewhere. And then again, imagine the tRNA, which I will simplify. This is going to take a lot of time to draw. So something like this, okay? Our tRNA is our friend that can understand the mRNA language. Why? Because in the first place, mRNA and tRNA are both RNA. So wouldn't it make sense that RNA has its codon right here, and then tRNA has its equivalent anticodon because it's also a nucleic acid anyway. And the goal here is that the correct tRNA for this particular codon should be able to have all of their bases matched with one another. That is to say, if I think of this as my first codon base, and the opposite one is the third anticodon base, they should match. The second codon and the second anticodon base should also match, as well as the third codon base and the first anticodon base. If this does not match, it means that this tRNA is probably not the tRNA intended for that codon. In that case, We need to find the proper tRNA. And this is an intrinsic way for us to proofread the translation process. Okay. Now, of course, since I did mention that our tRNA is that friend which can understand both languages, how would then we expect the tRNA to understand the language of proteins? And in that case, the answer lies with its first word, transfer. The goal of transfer RNA is to transfer amino acids. That's why, going back to the original structure of the tRNA, its end right here is actually an amino acid attachment site because the tRNA is designed in a way that it's supposed to hold amino acids. And in a way, it's kind of doing the job of delivering the proper amino acid that should match its corresponding codon, such that methionine is meant for the codon AUG. And then, of course, we know that we are bringing in the right amino acid because the codon and anticodon should match. Okay, there is an idea, by the way, called the wobble hypothesis, which states, although I will not anymore be discussing this too much in detail here, that there are possibilities that even if the third codon and the first anticodon base do not match, there's some kind of wobbling, loose movement that allows allowance for mistakes here. And it's kind of easier to understand why we need this if I have my 4 by 4 by 4 table, but that's the reason why I do not want to expound on this, probably in the future, but not now. But at least now, so far, we do know that other than the mRNA, we need, of course, our amino acids. That's how we make the proteins. And to bridge the mRNA and the amino acids, we have our tRNA here. So expect this to play a central role in the actual process. Now, of course, lest we forget the place or the region of the cell, well, where translation takes place is the ribosome. And even though probably in textbooks we would see ribosomes being portrayed as tiny dots, okay, if we zoom in, it's actually complicated because that dot that we often see is made up of many RNA and protein molecules condensed into, well, usually the way we see it, they are globular forms. And most functioning ribosomes are made up of two subunits or portions, wherein one is always larger. So I just call it the larger subunit here. And then the other one is smaller. Now, it depends on whether we are referring to a eukaryotic or a prokaryotic cell, um, uh, i- the way we label them. Okay. And the numbers you're seeing here are numbers that refer to their uh, S value, wherein S here stands for Svedberg. This is a unit of sedimentation, I believe. Not really my expertise, but the idea is whatever measurement was done and computation, we arrived at these numbers. For example, for the larger eukaryotic subunit, we call it 60S. So this is 60S for eukaryotes, 50S for the prokaryotes. For the smaller subunit, as you can see in the written part here, this is 40S for eukaryotes. You call this 30S instead for the prokaryotes. If you combine them, do note that the way the Svedberg units are acquired are not arithmetic, so are not arithmetically derived. So like you don't add 60 plus 40 equals 100, no? So you measure the sedimentation of the entire thing, which is not actually additive. So apparently if you combine the 60 and the 40 subunits, they will just give you a unit of 80S. So we call the entire thing here 80S for eukaryotes. For prokaryotes, instead, we call this the 70S ribosome. And another detail is that in the larger subunit, you have three major regions: E, P, and A. These actually have meanings. E stands for the exit region or the exit site. P stands for the peptidyl site. and then A stands for the amino acyl site. And just so you could have an idea for future discussion, I want you to think of translation as going from right to left. So actually, we enter the A first, and then we think of the process proceeding uh, uh, going to the left. So from A, we go to P, and then we go to E. Okay, which is opposite of what we usually do before, because we always go from left to right in the previous recordings. Okay, now, of course, the fact that we're performing a translation process means that we have to follow some rules, because just like, probably for people who have, for if you know multiple languages, you do know that you have to get acquainted with the rules of the new language you're learning, because otherwise the way you speak in the new language would actually sound bad. And therefore, in this case, the code, or the set of translation rules from RNA to protein, is called the quote unquote genetic code. And of course, just like learning a new language, if you do not observe the proper rules of this one, you will turn out having broken phrases and you know un hard to understand things. Okay, and basically, if you violate the genetic code, it's as if the proteins you're going to be making are either malfunctioning or useless. You want to follow these rules. So the five, most references list them as five. Some would add more, okay, but these are the five critical rules that I think you should know. First, the genetic code is almost universal, meaning that as long as we are talking about a living organism, be it bacteria, be it plants, fungi, humans, other animals, UUU, for example, is phenylalanine. AUG is methionine. Okay, so it's almost universal. And the way that I use almost here implies that there might be some violations, but they are rare. Two, the genetic code is said to be non ambiguous, or in other words, not confusing. So, how do we make this not confusing? Basically, it means that every codon that we have out of those 61 is equivalent to only one amino acid. So, the 61 codons do not have multiple meanings, right? Like, for example, if I say that UGG is equal to tryptophan, I mean it. It does not change. It's not like tomorrow, oh, the genetic code has changed, and maybe UGG tomorrow has been converted to threonine. That is not supposed to happen. In the first place, why would you even have a code wherein the codes are equivalent to different meanings? Wouldn't it suck that, for example, in the alphabet, you read this as G, and then tomorrow, instead of G, we now call this the letter O or P? That doesn't even make any sense. So it should stay like that. One codon will only mean one amino acid, so that we won't be confused. However, ironic that if we do this the other way around, one amino acid could actually have many codons. This rule is said to be the code of redundancy or degeneracy. For example, UUU and UUC are said to be redundant or degenerate codons because they are coding for the same amino acid, which is phenylalanine. And maybe it wouldn't be too absurd for you to think about it, because remember, how many amino acids have we discussed before that are part of the genetic code? Only 20, right? And how many codons are coding? Only 61. I mean, not only, but there are a lot. There are 61. So wouldn't it make sense that since I have fewer amino acids than I have coding codons, it's really bound to happen that some of those codons would repeat or be redundant for the same amino acid. So that justifies, I guess, in a very simple mathematical way, the rule of degeneracy. So it's like, if I'm going to simplify this 20 to 60, it's like on the average, there's going to be one amino acid um, having three codons. Of course, this is just an average. It doesn't mean that all amino acids have three codons. In fact, some amino acids have six codons coding for them. Some have four, some have two. In fact, there are even some exceptions. That is, there are some amino acids which only have a single codon. No repetitions, no redundant codons. For example, methionine is one of them because methionine only has the codon AUG for it, as well as tryptophan because tryptophan only has the codon UGG. And before I forget, since I already mentioned AUG, do note that other than being the sole or the only codon for methionine, AUG is sometimes called the start codon because we always find AUG at the very start of any mRNA strand that is being converted into a protein. Then we have rules 4 and 5, which mean which will we will use this sequence here below for. First, non-overlapping. The genetic code is said to be non-overlapping. What does that mean? It means that we cannot repeat the usage of one base for two or more codons. For example, let's say my first codon is 123, as in this one. The moment I used this, and this, I am not anymore allowed to use this for the next codon, any of them. So I cannot do like 123, 231. No, you cannot reuse this, or 123, 312. That would mean I have used this. Actually, when I say overlapping, you can see the blue lines here. It means that we repeat. For example, I go 123, 231, 123, 231, 123. The moment you see this, right, it's like an overlap. I used one twice here, I used two twice here, I used three twice here. That is not allowed. So how do you do it? So once you stop at three here for the first codon, your only choice would be the letters or the bases after. Now, in addition to that, I kind of think of 4 and 5 as a package deal. 5, the genetic code is also said to be commaless. By comma or some kind of punctuation, it's like a metaphor for us, indicating that we could skip on a base or two. For example, look at the purple lines here. This is a comma. For example, I'm done with 123. When I say a comma or a punctuation, or when I say the genetic code is punctuated, I skip on probably one or even two bases. So like 123, stop, 231, stop, stop, 123. These things are also not allowed. That means the moment you are done with your first codon, you cannot jump over any letter, be it 1, 2, 3, or 4 or 5. You have to proceed directly with the next one, such that after the first codon 1, 2, 3, a while ago I said you're not supposed to repeat them. But at the same time, you're not anymore allowed to repeat any of these three numbers. You're also not allowed to skip it. So like 1, 2, 3, 2, 3, 1, 3, 1, 2. That's overlapping. That's not allowed. But also 1, 2, 3, 2, 3, 1, 1, 2, 3. Those commas or pauses, or probably in music, you could call them rests, are not allowed. That's why you could see the underline below is the correct way of reading the genetic code. We just do it as kind of robotic, um, as possible. That is, you go straight, like as if it was a perfect tempo. 1, 2, 3, 1, 2, 3, 1, 2, 3, 1, 2, 3. Do not stop anywhere. Do not lag on any base, but do not skip any base also. By that, that's the way I think 4 and 5 should be combined. Sometimes I call 4 and 5 the rule of linearity. Okay, because it should just be continuous, okay, or continuity. But of course, I just use the terms that we find in references. Again, the genetic code should be read without overlaps and without commas. So We already have our mRNA here. I've already introduced to you our translator molecule, our friend right here, the site where the process takes place, and the rules that we have to abide to not come up with bad proteins. So in my next discussion, we will get to the actual translation process.